Today, we are going to explain the determination of sulfate in fertilizer. This will be presented by Irdina, Aina, Yasmin and Rashika. This experiment focuses on determining sulfate content in a fertilizer sample. Sulfur is an essential nutrient that helps support plant growth and development. Therefore, it is very important to determine the sulfate content in a plant to ensure proper quality control and nutrient management. To do this, we use gravimetric analysis. Gravimetric analysis is a quantitative analytical method based on mass measurement. It is widely used because it provides accurate and reliable results. In this method, the analyte is converted into an insoluble precipitate, which then can be separated and weighed. The mass of the precipitate is used to determine the content of the sulfur in the fertilizers.
This experiment uses gravimetric analysis, which is a quantitative analytical technique used to determine the amount of specific substance in a sample by converting it into an insoluble compound that can be isolated and weighed. In this experiment, the substance being analyzed is sulfate ion, which is present in the fertilizer sample. To separate the sulfate ion, an excess of barium chloride is added. The sulfate ion react with barium chloride to form barium sulfate, which is a white insoluble precipitate. The chemical reaction is barium chloride react with iron sulfate and form barium sulfate and iron chloride. Since the barium sulfate is insoluble in water, it precipitates out of the solution and can be collected. The precipitate then undergoes several steps, which is digestion, filtration, washing and drying until a constant mass is obtained. The mass of dried barium sulfate is then used to calculate the amount of sulfate in the fertilizer sample. Therefore, gravimetric analysis provides an accurate method for determining the sulfate content in fertilizer.
First, the fertilizer samples was dissolved in distilled water to ensure that all the sulfate ions were present in the solutions. Then, barium chloride was added, causing a white barium sulfate precipitate to form. Next, the mixture was filtered using filter paper to separate barium sulfates from the solution. It then washed with distilled water to ensure there is no remaining impurities. Finally, the precipitate was dried and allowed to cool before weighing. The mass of the precipitate is then used to calculate the content of the sulfate in fertilizer sample.
Here the example result that we use for our presentation. Mass of beaker is 111.5 gram. Mass of beaker with fertilizer is 113.9 gram. So mass of fertilizer with beaker minus with mass of beaker to get mass of fertilizer, which is 2.09 gram. Mass of filter paper is 1.38 gram. Mass of filter paper with barium sulfate is 3.16 gram. So mass of barium sulfate with filter paper minus with mass of filter paper to get the mass of barium sulfate, which is 1.78 gram.
Moving on to the calculation, our primary objective is to determine the percentage of sulfate ion present in our fertilizer sample. The basis of this calculation relies on the precipitation reaction shown at the top. Barium chloride reacts with the sulfate ion to form a solid precipitate, barium sulfate, and chloride ions. First, we need to calculate the number of moles of barium sulfate. To get the actual mass of the precipitate, we subtract the mass of filter paper and barium sulfate and mass of filter paper. Dividing this net mass by the molar mass of barium sulfate, which is 223 gram per mole, we find that we obtain 0.00763 of barium sulfate mole. Next, we look at the stoichiometry of our balance equation. The reaction shows molar ratio between barium sulfate and the sulfate ion is 1:1. Therefore, the number of moles of originally present in the fertilizer is exactly equal to the moles of barium sulfate produced, which is 0.00763 moles. Now that we know the moles of sulfate, we convert it back into gram to find its mass. We multiply 0.00763 with the molar mass of sulfate ion, which is 96 gram per mole. The mass of sulfate ion is 0.723 gram. Finally, we calculate the total percentage of sulfate in the original fertilizer sample. We take the mass of the sulfate we just found and divide it by the net mass of the fertilizer used, which can be calculated by the subtraction of mass of fertilizer and beaker and mass of beaker. and we multiply with 100%. We conclude that our fertilizer sample contains 30.5% sulfate.
So this slide shows the benefits of sulfates in daily life, which can be divided into three categories, which is health and wellness, personal care and hygiene, also household cleaning. The first one is the health and wellness, which is we have magnesium sulfate, also known as Epsom salt, that act as an osmotic laxative that creates pressure that stimulates the intestinal muscle. Also, we need to use it with caution because it can lead to diarrhea or dehydrations. And then detoxification measure cleansing process that cause sulfations. For example, many toxins and drugs liver chemically transform these compounds to make them water soluble that has three phases, which is biotransformation, conjugation, and eliminations. And then we move to personal care and hygiene, which we have exfoliation, facial body scrub that can remove dead skin cells and excess oil to lead for a skin smoother and cleaner. Also can act as an cleansing agents that break down excess oil and impurities. And then deep cleaning also can help to remove dirt, oil, sweat, and bacteria from hair and skin using soap, body washes, or shampoo. Also can reduce excess sebum that can clog pores. And then lastly, household cleaning, water softening and laundry detergent, which have sodium sulfate that have a effectiveness cleaning in hard water, also act as an strong cleaning agents that can remove dirt, oil by breaking down the grease, and kills and remove germs when dirt and oil remove. Germs that attach to the surface also will be washed away. So sulfates helps water penetrate surfaces deeply and maintain cleaner clothes and healthier living environment.
There are safety implications that we need to consider while doing this experiment. Number one, wear the complete personal protective equipment (PPE) such as lab coat and enclosed footwear before do the experiment. Number two, barium compound is toxic, so we need to wash our hands with soap and water before leaving the lab. Number three, cool the precipitate before weighing to prevent the moisture absorption. Number four, pour the solution slowly into the filter paper to avoid loss of precipitate during filtration, which can reduce the final precipitate weight and affect the accuracy.
So in conclusion, the determinations of sulfates in fertilizer was successfully conducted using the suitable chemical analysis methods. The experiment confirmed the presence of sulfates ions in the fertilizer sample and demonstrated the importance of sulfate as an essential nutrient for plant growth. Sulfur helps in the formations of proteins, enzymes, and chlorophyll, which are very important for healthy development and higher agricultural productivity. The experiment also provided a better understanding of analytical techniques used to identify and measure sulfate compounds. Although the minor experimental errors such as inaccurate measurements, incomplete precipitations, or loss of sample during filtration may have affected the accuracy of the results, the overall objective of the experiment were achieved successfully. This experiment highlights the importance of sulfates analysis in ensuring the fertilizer quality.